So retinopathy is a major complication of diabetes, and it's a leading cause of blindness. As Ram mentioned earlier on, changes in the vasculature in the retina are an important component of diabetic retinopathy. And these changes include the loss of pericytes, the loss of endothelial cells that lead to the formation of the so-called degenerative capillaries, which in essence represent tubes of basal membrane that lack blood flow. And these areas of localized ischemia in the retina are thought to contribute to neovascularization, which is one of the features in later stages of diabetic retinopathy. There is ample evidence that a chronic low-grade inflammation plays an important component in the development of diabetic retinopathy. For example, it has been shown that both in humans as well as in mice with diabetic retinopathy, there's increased leukostasis, which means an increased number of leukocytes that are adherent to the capillaries. In addition, there's evidence of upregulation of ICAM1, which is an adhesion molecule that is key for driving enhanced leukostasis. It's also known that patients with proliferative diabetic retinopathy have increased levels of the chemokine MCP1 in the vitreous, and that these levels of MCP1 correlate directly with disease activity. NOS2 has been shown also to be critical for driving diabetic retinopathy, both in humans as well as in rats with this disease. The levels of NOS2 are increased, and NOS2 deficient mice that are made diabetic have diminished the histopathological changes of diabetic retinopathy. And finally, pro-inflammatory cytokines such as IL-1 and TNF-alpha have been linked to diabetic retinopathy. While there are major advances in the understanding of the pathogenesis of this disease, it is still not very well understood how these pro-inflammatory responses are regulated. CD40 is a member of the TNF receptor superfamily that was described initially on antigen presenter cells. Its major ligand is CD154 expressed on activated T cells. Subsequent work revealed that CD40 can also be expressed by some non-hematopoietic cells. CD40 plays a dual role in disease in humans. CD40-CD154 interaction is key for resistance against many pathogens. Thus, humans that have a defect in this signaling pathway are prone to opportunistic infections. However, it is also known that CD40 can drive diseases that have an important inflammatory component. We've recently demonstrated that primary retinal cells in humans as well as in mice express CD40, and these cells include retinal endothelial cells, MULLER cells, pericytes, and microglia. And this led us to ask the question of what the role of CD40 is in the development of diabetic retinopathy. So we began by looking at what happens to the expression of CD40 in the retinas of diabetic mice. So black 6 mice were made diabetic by injection of streptozotocin, and at 2 months after diabetes, the levels of mRNA of CD40 in the retina were quantitated by real-time PCR. And compared to the expression of CD40 in control non-diabetic mice had a significant increased expression of CD40. Then we asked what was the role of CD40 in leukostasis. So here we took black 6 mice and CD40 deficient mice, and again they were made diabetic. And the blood glucose, glycated hemoglobin, and body weights were similar in both diabetic black 6 mice as well as CD40 deficient mice. And then at 2 months after diabetes, we quantitated the number of leukocytes that were adherent in the capillaries. And as expected, diabetic black 6 mice had increased leukostasis compared to non-diabetic black 6 controls. However, this leukostasis was significantly impaired in CD40 deficient mice, indicating that CD40 promotes leukostasis in the retina. I mentioned that ICAM is key for driving leukostasis, so we obviously wanted to know what happens to upregulation of ICAM in CD40 deficient mice. So here again, black 6 mice and CD40 deficient mice were made diabetic, and here the expression of ICAM1 was measured by real-time PCR and is expressed as the fold increase compared to the expression of ICAM1 in non-diabetic controls. So in diabetic black 6 mice, there is a 5-fold increase in ICAM1, and this upregulation is significantly blunted in CD40 deficient mice who were made diabetic. NCP1, as I mentioned before, is a chemokine that has been linked to diabetic retinopathy, at least in humans. And as we can see here, the black 6 mice also have increased levels of MCP1 mRNA, and there is no upregulation of MCP1 in diabetic CD40 deficient mice. The development of acellular capillaries, so degenerative capillaries, as well as pericyte growths, are the signature lesions of diabetic retinopathy. Here we took black 6 mice and CD40 deficient mice that were made diabetic, and at 8 months of diabetes, we quantitated the number of acellular capillaries. And as expected, there's evidence of capillary degeneration in diabetic black 6 mice. However, diabetic CD40 deficient mice were protected from capillary degeneration. Similarly, parasite ghost, which is evidence of loss of parasites, occurred in diabetic black 6 mice, but it did not occur in CD40 deficient mice. And these data demonstrate that CD40 is key for the development of diabetic retinopathy. Then we moved to in vitro experiments to further confirm the relevance of CD40 for regulation of these pro-inflammatory responses in retinal cells. As I mentioned at the beginning, human primary retinal endothelial cells and Muller cells express CD40. When we treated these cells with a ligand for CD40, CD154, there was upregulation of ICAM1 and MCP1, but this was moderate, not surprisingly because under basal conditions, cells express low levels of CD40. So to further confirm that CD40 can indeed control expression of these pro-inflammatory cells in pro-inflammatory markers in retinal cells, we transduced human primary retinal endothelial cells and Muller cells with a retroviral vector that encodes CD40. When these cells were exposed to CD154 as opposed to control, there was marked upregulation of ICAM1 both in primary human endothelial cells as well as in primary human Muller cells. When we look at the production of MCP1, we also found that CD40 stimulation triggered MCP1 production by both human endothelial cells as well as Muller cells. And not shown here is that we find similar results with primary human parasites. We then examine whether CD40 can also control NOS2 expression. So this is data obtained using human primary Muller cells that were treated with CD154 control, and we can see that in response to the ligand, there is some upregulation of NOS2 by Western. These cells had a basal expression of CD40, which, as I mentioned at the beginning, is normally at low levels. So to further confirm that CD40 can trigger upregulation of NOS2, we transduce these cells with a retroviral vector that encodes wild-type human CD40, and in response to CD40 stimulation, there was a marked upregulation of NOS2. Not only NOS2 was upregulated by Western, but we also were able to find detectable nitric oxide by Grice reaction, and we also found similar results when we tested a human microglial cell line that showed also NOS2 upregulation as well as ANO production in response to CD40 engagement. The development of cellular capillaries is thought to be due to the loss of endothelial cells by cell death that has features of apoptosis. So we wanted to know whether CD40 could drive apoptosis of endothelial cells. We found that if we were to engage CD40 directly on endothelial cells, this would not lead to apoptosis of these vascular cells. So then we proposed that perhaps what was happening is that if we were to stimulate CD40 on glial cells, this would lead to death of apoptosis of bystander endothelial cells. So we took glial cells, which were transduced with a retroviral vector that expresses CD40, but this vector also expresses EGFP, so more than 95% of the cells were EGFP positive. And these cells were incubated with primary human endothelial cells at a ratio of 4 endothelial cells per 1 Muller cell. And then we added CD154 to this system. And we assessed apoptosis of endothelial cells by determining expression of annexin 5 and 7-AAD. And as we can see here, in response to CD40 stimulation, there was increased levels of apoptosis in endothelial cells. And in one experiment so far, this increased apoptosis was blocked by adding the NOS2 inhibitor Elnyl. So basically, we have shown that CD40 is a new regulator of diabetic retinopathy that is key for the development of this disease, and that CD40 can control many of the pro-inflammatory responses that are linked to the development of diabetic retinopathy. So this cartoon summarizes the model that we proposed. It has been shown that diabetic patients with retinopathies have high levels of CD40 in the serum as a soluble protein. And also it is known that both in humans as well as in animals, in the retina of diabetic individuals, there is evidence of thrombi with platelets. And platelets are also a key source of CD154. So either of these sources of CD154 would then interact with CD40 expressed on endothelial cells, pericytes, microglia, and Muller cells. This will lead to upregulation of ICAM1 in endothelial cells, which is key for leukostasis. There are data from other laboratories that have shown that leukostasis drives capillary degeneration by leading to apoptosis of endothelial cells. However, it's important to point out that leukostasis has a partial role in driving capillary degeneration, which indicates there must be another mechanism by which endothelial cells are dying. And we are proposing that CD40 stimulation of glia will lead to apoptosis of endothelial cells through NO production. And we also have data that I'm not showing today indicating that CD40 also triggers high levels of prostaglandin E2, which can also be pro-apoptotic. So the people in the lab that did the work with Jose Andres Portillo, postdoc in the lab, and Genevieve Kenka, and this work was done in collaboration with Tim Kern and Ram Nakaresh. Thank you very much.